Welcome back to The Explainer. Today, we are cracking open Chapter 9 to demystify the ultimate recipe book, the chemical building blocks of life itself. We're going to look at the absolute foundation of what makes a living thing, well, actually living.
Let's kick things off with a pretty massive question. What exactly is the difference between living tissue, like your body, and a clump of non-living earth? I mean, are you really just made of dirt? Well, here's the staggering cosmic truth. In absolute terms, there's literally no difference in the types of elements found in the Earth's crust versus the elements in your body. You've got sodium, oxygen, magnesium, it's all there in both. The real difference, it all comes down to the relative abundance of carbon and hydrogen. You just have a massively higher proportion of carbon and hydrogen compared to the rocks outside your window.
So, here's our roadmap for solving this mystery together today. We'll cover the chemistry of life, how we extract the ingredients, primary and secondary metabolites, the mighty macromolecules, protein architecture, and finally, enzymes. Let's dive in.
Starting with part 1, the chemistry of life. To figure out what we're actually made of, we have to zoom way down to the atomic level. If we do what's called a destructive elemental analysis, basically taking a tissue, drying out all the water, and burning it, we can examine the ash that's left over. And what we find is that the true signature of living organisms isn't some magical, undiscovered element. It's just a uniquely high concentration of carbon and hydrogen forming complex biomolecules. Which brings us to part 2, extracting the ingredients. You might think isolating these tiny chemical components requires incredibly high-tech gear, right? Actually, it's a surprisingly physical, almost low-tech process. Scientists just take a piece of living tissue, grind it up in trichloroacetic acid using a mortar and pestle, and make a thick slurry. Then you just strain that slurry through cheesecloth. It creates this beautiful separation. You get an acid-soluble pool of liquid containing thousands of tiny organic compounds that filter right through, and an acid-insoluble fraction left behind in the cloth that holds all the heavy cellular machinery. Let's look closer at that liquid in part 3, primary and secondary metabolites. We divide the thousands of tiny organic compounds in that acid-soluble pool into 2 main groups. First, primary metabolites. These handle your normal everyday physiological processes. Think essential amino acids, simple sugars, and lipids that keep the cell running. But then we have secondary metabolites, and these are fascinating. Found a lot in plants and fungi, these are compounds like rubber, antibiotics, toxins, and scents. We might not always know their exact physiological role for the host organism, but they have incredible ecological importance and, frankly, massive benefits for human welfare. To put a visual to it, these are some of those fundamental primary metabolites. You've got rings of sugars, the structures of amino acids like glycine and alanine, and complex nucleotides. They form the base of that acid-soluble pool. These are the true micromolecules of life, and they typically weigh anywhere from just 18 to about 800 daltons, which, by the way, is just the standard unit we use to measure atomic mass. All right, part 4, the mighty macromolecules. What about the stuff that didn't dissolve? The crucial thing to understand here is the sheer scale. Macromolecules, like proteins, the nucleic acids that make up your DNA, and large polysaccharides, are absolutely massive. While those soluble micromolecules we just looked at maxed out around 800 daltons, these giant polymers have molecular weights largely exceeding 10,000 daltons. 10,000! They are the undeniable heavy lifters of biology. When you actually break down a cell's average composition, it's kind of wild to realize that 70 to 90% of it is simply water. After water, proteins form the very next largest chunk at 10 to 15%, followed by nucleic acids and carbohydrates. But there's a super interesting exception to our size rules: lipids. Technically, lipids are small molecules weighing well under 800 daltons. So why on earth do they show up in the heavy acid-insoluble fraction when we strain our tissue slurry? It's because lipids form the cell's physical membranes. When you grind up the tissue, those membranes shatter into tiny insoluble pieces that get tangled and trapped up in the cheesecloth right alongside the giant macromolecules. That takes us right into part 5, the architecture of proteins. We know proteins make up a huge chunk of our solid mass. To give you a real sense of scale here, collagen is the most abundant protein in the animal world, physically holding your body together. But the absolute king? That's rubisco. It's a plant protein essential for photosynthesis, and it is, without a doubt, the most abundant protein on planet Earth. The secret to a protein's power is its architecture. They don't just stay as flat strings. They climb a very specific structural ladder. The primary structure is just the linear sequence of amino acids. Simple enough. Then they twist into a secondary structure, kind of like a spiraling helix. Next, that long thread folds upon itself like a hollow woolen ball, creating an incredibly complex 3D tertiary shape. And finally, they can lock together into quaternary structures, where multiple subunits fit together perfectly like puzzle pieces to make functional structures like human hemoglobin. And that intricate 3D folding leads us directly to our final section, enzymes, the cellular workers. Because proteins fold upon themselves in such complex ways, the chain crisscrosses and creates very specific crevices or pockets. We call these active sites. A chemical substrate fits perfectly into this pocket, basically turning the protein into a highly efficient biological catalyst. They drive the chemical reactions of life at absolute breakneck speeds. Almost all enzymes are proteins, and they're literally the active workers keeping the cell alive. But here's the catch. And really, this is a profound difference between living biology and non-living chemistry. Inorganic catalysts in a lab thrive in extreme heat and high pressures. Biological enzymes, on the other hand, are incredibly fragile. Because their whole function relies entirely on that perfectly folded 3D shape, they get damaged and unraveled at high temperatures, usually anything above 40 degrees Celsius. This is exactly why a high fever is so incredibly dangerous for you. Your cellular workers literally begin to lose their shape and stop working. So as we wrap up this explainer, I want to leave you with a bit of a mind-bending thought. Everything we've discussed today is just basic chemistry. So how do these simple, completely inanimate carbon compounds orchestrate themselves to create the magnificent, conscious life you are experiencing right now? It's the ultimate puzzle of Chapter 9, and really, of biology itself. Thanks for joining me on The Explainer. Until next time, stay curious,